If you would have asked me 5 years ago what the on-trend brewing thing would be in 2021, I never would have guessed that hard seltzer water would be so popular. Laugh all you want, but hard seltzer is a fantastic day drinker and likely isn't going anywhere in the near future. Imperial Yeast's new seasonal strain, W04 Paramount, can help brewers get the most out of their seltzer fermentations. A clean and aggressive fermenter, Paramount will produce an excellent seltzer with low fusel alcohols, and it's produced in a gluten-free medium. If you've tried making seltzer with standard ale or lager strains, you know the struggle, and Imperial Yeast is here to help with W04 Paramount. Check it out at imperialyeast.com. Welcome to the Brew Lab. The brewing industry is replete with new technologies and tools that reduce time and costs or help brewers go in new directions or explore new places. Utilizing those new tools is great, but there's often something to be said for continuing traditional practices that, despite being maybe more time-consuming, might generate new or novel results that move brewers into new spaces. I'm your host, Cade Job, and today in the lab I'm speaking with Richard Preiss of Escarpment Laboratories about some recent successes they've had utilizing traditional yeast breeding techniques to create novel, new strains of yeast. Now, we've done a couple of episodes on how genetic engineering or genetic modifications can be used to modify yeast, but we've only done one episode on traditional yeast breeding, which was way back almost 3 years ago in episode 6 with Matt Winans. Using traditional methods like Richard and the team at Escarpment have been using, they've been able to further push the boundaries of what we're able to accomplish with yeast and have had several recent successes. So today we'll talk about what do we mean by traditional yeast breeding methods? How do we come up with an idea? How do you make a yeast hybrid? And once you've made a hybrid, how do you make sure that it does the thing that you want it to do? Or in some cases, in the case that it doesn't do the thing that you want it to do, finding something that's still really cool about it in its own right that might make it a strain that you would want to release. Hint, hint, foreshadowing. But I digress. I've been super impressed when I've talked to Laura Burns from Omega Yeast and Nick Harris from Berkeley Yeast talking about their GMO products, and I've got to say I'm equally impressed by the work coming out of Escarpment. So I'm really excited to talk to Richard today about that, more on the successes that they've had with traditional yeast breeding later in the show. If you haven't yet signed up to become a patron, please consider doing so. By becoming a patron of Brewlosophy, you get awesome rewards like access to Brewlosophy contributor recipes, new discounts each month to YakimaValleyHops.com, and for $3 a month, access to a monthly live Q&A session with a special guest from the brewing industry. Becoming a patron is super easy. Once you've contributed at that $3 level or higher, you get access to the private Patreon Facebook page where all of the previous Q&A sessions are available. I've said it before, but guests like Vinny Chilurzo, John Palmer, and the Brewlosopher himself, Marshall Schott, they're all available on that private Patreon Facebook page. And it's just $3 a month. There are so many more guests also to choose from. All the information that you need to get access to this awesome resource is available at patreon.com/Brewlosophy. So please go check it out. Thank you to everyone who's left a rating or review of the show. We're over 100 ratings on Apple Podcasts, which is awesome. And if you haven't yet left a rating or review on your podcast service of choice, I'd really appreciate it. Take a second, let us know what you think of the show, and help others like you find us. Feedback this week is from listener Scott, who says, Cade, I really enjoy and appreciate the Applying the Science episodes. I'm fairly well read in the scientific literature, medicine, but some of the earlier episodes were very technical and hard to follow. The addition of the Applying the Sciences episodes has been a game changer for me and really ties together the material discussed by the researchers who contributed to the primary literature. Thank you. Enjoyed your master's thesis defense episode as well as the follow-up episode with Tom Schallhammer about hop creep. It's interesting to know that many different plants, including hops, do have genes for the amylase enzyme. However, I read somewhere that hops also have limit dextrinase enzyme, which would make sense as it could break down the residual dextrins in wort in the hop creep process. I may have missed this in your episodes, but did you discuss limit dextrinase in hops? Do hops have the gene for limit dextrinase? And if so, is this gene expressed in hops? As an aside, I've definitely seen the effects of hop creep on the homebrew scale. My first West Coast style double IPA hopped with 4 ounces of hops at a 5-gallon batch. ended at a stable terminal gravity, resulted in new airlock activity. And after 4 days and removing the hop bag, I noticed that the gravity had dropped by 4 gravity points after dry hopping. At that time, I'd not heard of hop creep, so I initially thought this could be an infection. Interestingly, I entered this beer into a large regional competition, and the judges detected a low level of diacetyl. I think this was hop creep. Appreciate everything that you do, Brewlosophy patron Scott. Hey, Scott, thanks for that. Thank you also for being a patron. And thank you for the anecdote, first of all, about hop creep in your home brewery. It certainly sounds like that's what you got, right? You added the hops and then saw a decrease in gravity and maybe even a little bit of diacetyl. So yeah, I think that's exactly what happened, or at least it sounds like it to me. But I'm curious now, do you do anything differently to avoid hop creep? I'd be interested to hear that. I talked with Jordan a little bit about it on the Applying the Science episode, as you probably know. But what are home brewers doing to avoid hop creep? I'd like to know a little bit more on that. You also asked about limit dextranase. Yes. So your question is, do hops have limit dextranase? Is the gene expressed? And I don't know that I've seen it published in a paper. I looked through the research briefly in answering this question, but I didn't see anything, certainly nothing like Matt Cottrell's work with the amylases, where he just sort of, methodically shows that amylase genes exist in hops. I haven't seen a similar study with limit dextranase, but I think you're totally on track there. And this, of course, this is totally me speculating, but I think limit dextranase has to be there. It just makes sense from a starch chemistry perspective. It would make sense about the residual dextrins that you're talking about. And so, I'm not a betting person, but if I was, my money would be that someone, if they do the work, will confirm that limit dextranase exists in hops. Expression is a whole other animal, and even amylase, I don't think, has been shown to be a gene that's expressed in hops. I think the pieces are there, but again, nobody's done the work yet. And I hope someone will do this. And I hope that if they do the expression study with amylases, that they also add limit dextranase in too, and see if those genes exist and if it's also expressed. So I would love to see that as well. I think that's a little bit of a hole in the literature about the gene expression. We know that the genes exist, but we don't know if they're actually expressed yet, or at least hasn't been scientifically proven. Hop creep exists, and so we think that the genes are being expressed, but nobody's shown it in the literature, at least as far as I'm aware of. So Scott, thank you for the questions, and thank you for the anecdote about hop creep. All right, I'll be right back after this break talking about traditional yeast breeding practices with Richard Preiss. One of the biggest improvements to my brewing practices was the upgrade to stainless steel, and Delta Brewing Systems offers some of the lowest prices on high-quality stainless gear, like the Firm Tank, which holds 8 gallons or 30 liters of wort, comes with a domed lid to reduce the chances of a messy blow-off, and it can hold up to 4 psi of pressure. Delta Brewing Systems also has their own line of brew kettles, as well as one of the lowest-priced all-in-one electric brew systems out there, and their prices are remarkably affordable. If you're in the market for legit stainless gear that won't break the bank, you've got to check out deltabrewingsystems.com. We all know that designing recipes is really fun, and doing it well is so much easier with good software. We at Brewlosophy recently made the switch to Brewfather, and honestly, y'all, we could not be happier. Brewfather utilizes the latest technology to bring you the most robust cloud-based recipe design software that can be accessed anywhere, on your phone, tablet, desktop, and even offline. It also works seamlessly with numerous third-party devices to make it easier to monitor every step of your brew. I know change can be difficult, but trust me when I say you need to go to brewfather.app today to try it out for yourself. That's brewfather.app.
The mighty and resilient Merrimack River, carving through the communities of our great region. My name is Linda Lorden, proud president of Merrimack County Savings Bank. And like the river that serves as our namesake, we're a constant yet ever-changing presence. Because to us, it's bigger than banking. It's about powering communities and putting people first. It's about knowing where you came from and where you're going. That's Merrimack style. Visit us at themerrimack.com.
Yeast carries a heavy load in beer production, from the brass tacks of making alcohol to the flavor compounds produced during fermentation. We've done a couple of episodes on genetically engineering yeast to solve brewing-related issues like diacetyl or haze, or to increase flavor like the thiolized episodes, but we've yet to dive into detail about traditional breeding practices. Joining me in the lab today to discuss some recent successes from traditional yeast breeding is Richard Preiss of Escarpment Laboratories. Richard, welcome back to the Brew Lab.
It's good to be back, Cade. Really looking forward to this.
It's been far too long, right? I mean, I just had Tom Shellhammer on about 2 weeks ago, and he was episode 1, and you were episode 31. So it's been like the first year of the show, and now we're in year 4, and wow, it's the first time that you've been back. But it's been too long. And on that episode, of course, we were talking about the genetic lineage of Kveik strains, and I think on that episode you mentioned that because of the breeding conditions that Kveik strains have, they actually evolved into genetically distinct strains, things like trehalose accumulation and allowing them to ferment faster. And that's kind of cool, right? That evolution is what makes new strains naturally.
Yeast is constantly evolving, and it's constantly responding to the environment that we put it in. And yeah, I mean, Kveik's a great example. I mean, that was years ago, and we've done more research, and we still don't know exactly how it works, and we're still figuring things out and where it came from. So research does move slowly, but it does move. It does move over time.
It moves. Well, and I just think it's cool too that we can discover this thing 175 years ago or so and still to this day have new things to research. I just think that's fun.
You got it. It's I think I'll spend my whole life trying to figure out yeast.
Yeah, you probably will. That's right. Well, of course, I think everybody here knows who you are, Richard. Of course, you're co-founder of Escarpment Laboratories. You had a Master of Science in Molecular and Cellular Biology at the University of Guelph in Canada. So what have you been up to at Escarpment since we last spoke?
Yeah, that was a long time ago. So I think a whole pandemic happened. So helping a lot of brewers through that the ups and downs of the pandemic. Especially through towards the tail end of that, and how do we operate efficiently and innovate in inflation and all those fun things. But that's been a good challenge. Through all of that, we've been making and launching new yeast strains. Really trying to have our own perspective on yeast development and, make new strains available to people and just give them more options, more colors to paint with, so to speak. And then, of course, continuing all of our yeast research as well. So we're constantly trying to understand beer yeast better and to apply what we learn to hopefully help people make beer better. And that's still very much happening. We're still learning things and, trying to share what we learn all the time.
Yeah, which is actually where you and I reconnected a few weeks ago at the World Brewing Congress up there in Minneapolis. You guys were showing off a success of traditional yeast breeding practices, which we'll talk about in a little bit. But that's Pomona, right? A new yeast strain that you guys have developed and is available now for brewers to brew with.
That's correct. Yeah, it's Pomona is we've released it as a partnership with Lallemand. So that's really cool because it's now available across the world. Although the first batch sold out really fast. So the second batch, when this podcast airs, it should be, flowing all over the world and brewers should be able to use it. So that's really exciting. I'm actually drinking a beer made with Pomona right now. So I wanted to give a shout out to Short Finger Brewing just down the road in Kitchener. They made this cool beer called Cloud City. It's actually a few months old at this point, but it's tasting super fresh. It's a double dry hop pale ale with Nectaron Motueka. I do got to say, Nectaron and Pomona is a really nice combo. So there's a pro tip right off the top for the brewers listening. Try those two things together. You'll probably make a pretty good beer.
Yeah, get some of those New Zealand hops and throw them in there with Pomona. That sounds cool. Well, awesome. Yeah. So again, you and I met, or reconnected again at the World Brewing Congress, where we were both talking about all of the fun things that brewing researchers are up to. And Pomona again was, you guys were showcasing that yeast, and we realized you and I had talked about doing an episode on traditional yeast breeding like almost 2 years ago. And for whatever reason, both of us just got busy and forgot to do it. But here it is, and we finally got around to it.
We're ready. Yeah.
We're ready. Well, okay. So, in light of most of the recent episodes that I've had on yeast have been dealing with genetic engineering, right? Or GMO yeast. And so, in today's world of these crazy tools like genetic engineering, which often sounds like we can kind of do whatever we want with yeast, I know there are limits to it, but is there still a role for traditional yeast breeding?
I think so. Yeast breeding can help you hit targets that are not achievable with engineering, especially if the system that you're trying to alter isn't well understood. So, in the case of brewing yeast, some of the traits of the yeast that I think are interesting to modify or change are complex. It's not just like one gene turns that trait on or off. And so that's a case where something like breeding can actually be really advantageous. So if you're looking to improve a yeast's alcohol tolerance, or if you're looking to increase the expression of certain flavors that are what we call polygenic, it's like multiple genes that influence it, or other complex traits like flocculation, the way that yeast flocculates, it's a bunch of different genes interacting with each other. Then that's a case where breeding can have some advantages over more targeted engineering.
Yeah, that makes sense, right? And I think it was sort of explained to me that, like, yeah, targeted engineering can help deal with specific problems. And like you said, if there's a target gene that makes sense that you can modify or change, then yeah, genetic engineering works. But I remember way back, Matt Winans was talking about, who's now with Fermentus, and Chris Willig, who's a genetic scientist here at OSU. They were talking about how traditional breeding is really like the new front is like the discovery phase, the explorational science. It's like the brave new world, boldly going where no one's been before. Whereas genetic engineering is targeted. It's so hyper focused. It's like, let's look at this specific gene and see what happens when we play with it. Is that sounds like a lot like what you're talking about here with traditional breeding. Traditional breeding is this discovery and explorational science.
Well, there's that whole joke about engineering that, every engineering problem can be solved with duct tape or WD-40, right? So, engineering is a lot more targeted, right?
Yeah.
And it's not necessarily as into scientific exploration. Not to say that it's a bad thing. I think it's a great way to achieve a certain target. But I do agree. With breeding, you don't know what you're going to get, so there is a little bit of a discovery, a mystery aspect there. You can take two strains and try to breed them together and get a result that you completely didn't expect, for better or for worse. And we've seen those interesting results. So it really just comes down to, like, what do you understand the system, and what do you have patience for?
Yeah. Oh, yeah, that's true. And we're going to talk about patience here soon, too, because as I understand, some of these techniques do require that patience. But before we leave this topic, thinking about like genetic engineering versus traditional breeding, it is interesting to think about this as discovery and explorational, because it's like you said, it's like if you understand the system and you're trying to solve a problem, you can you can use these traditional techniques to solve those issues. And like an example might be, okay, yeah, sure, like the Haze gene, right? That Omega Yeast and Laura Burns and her team has discovered. That's one gene, and so you're able to modify that gene, and you can knock out that Haze component. But a lot of these things are complicated. A lot of these things have different uses, right? The yeast, again, doesn't care about your beer. All they care about is surviving, right? And living their life. So when they do something, they do it for a survival purpose. They're not necessarily, or making a metabolite or something, or finding something that they need in order to reproduce and live their life. And that's kind of where I see this traditional breeding really helping, is because, okay, let's say we've got a brewing problem that we need to solve, put the yeast in that and see how the yeast solves it, and see if they do solve it, kind of thing, right?
Yeah, you got it. And like, that's the kind of stuff that inspires me, is like having an empathy for the organism, and being willing to meet it on its terms and understand what makes it tick. That's the kind of stuff that excites me. At the end of the day, we develop yeast, so we have to end up with a product backed by science. But like, that's really the cool thing, is with some of those methods, yeah, you are kind of at the behest of the yeast, and maybe your idea of how to improve or change a yeast may not actually result in the thing you wanted, and you get some completely different result because that's what worked for the yeast. Because yeah, they don't really care about us. They just, care about consuming sugar and making cells and replicating, as efficiently as possible in most cases.
Yeah, exactly. Exactly. And this is kind of where I see traditional breeding and genetic engineering kind of as like parallel paths, right? Is like they both do beneficial things. It's not like to say that traditional breeding is better than genetic engineering, or at least that's my opinion. I don't know if that's your opinion, but these two things it's like all the tools in the toolkit. When we're brewing, we're adjusting things like mash temperature or mash tun size or, salt additions or pH. And it's kind of the same thing with yeast, right? You've got different tools in the toolkit. And so I see traditional breeding as like a really discovery sort of explorational tool, like an understanding. And even, you can still be targeted with traditional breeding, which we'll get to. But then you have, genetic engineering and genetic modification where we can play around and use modern tools to also achieve outcomes, so it's kind of parallel paths going the same way or going in the same direction, making great yeast.
Yeah, and I mean the people who work with me probably will, make the point that I can get a little galaxy brained with this stuff sometimes. But yeah, I mean you can go in every direction. You can stack methods on top of each other. It really is like choose your own adventure when it comes to, how do you improve a yeast or make a new yeast or make it better, right? You can use all of the tools in the toolkit to make improvements.
Yeah, I like that, galaxy brained. I'm going to use that. I think that would probably describe me too. I'm sure there's some listeners of the podcast out there going, yeah, Cade's pretty galaxy brained too sometimes. But yeah, okay. So we talked about like, using or yeast, what it's doing, right? Again, it's the main workhorse. It's creating alcohol and CO2 for us, but it's just out there living life. When you're breeding a new or trying to breed a new yeast, how do you start? Like what's the beginning of the process for you when you're looking at, okay, we want to come up with a new yeast? Is there like a big whiteboard in Escarpment Labs with like, here's an idea?
Kind of. I mean, first of all, we do think that, there's room for improvement in pretty much every yeast and every like category of yeast and beer style, simply because the yeasts that we're using to make beer are all pretty much all like 40 years old plus and haven't really changed since then. Like if you look at the yeasts that are commonly used in the industry, a lot of them were banked in like the 60s or 70s or 80s and haven't changed since then. So like it's kind of low-hanging fruit and there's a lot of room for improvement. And then where we tend to start is, as opposed to a whiteboard, you can almost look at it as like a map of, can we take a bunch of different traits of different strains and build a map or build it can even be as simple as just like four quadrants of, what can they do, flocculation, flavor, whatever it is, right? Can we build a map and can we identify where there's gaps in those combinations of traits in the yeast where fixing those gaps would be useful to brewers? So an example that I can use is there's actually not a whole lot of strains that are in terms of conventional strains that are really, really good at both flavor production, biotransformation, as well as fermentation efficiency, and that don't have like critical flaws in terms of off flavor production or nutrient requirements. Like you might have a strain that's like awesome at biotransformation, but it produces a ton of diacetyl, and you need to like go fix that or use enzyme or something like that. So there's a lot of these like interesting gaps, and that tends to be where we look to say, okay, can we, design a process to create a strain that fills in that gap?
Yeah, I like that, and I've read on your website too that that says like you guys believe that the vast majority of conventional brewing yeasts have critical weaknesses and flaws that can be overcome using scientific tools. That sounds like exactly what you were just describing.
Well, it's kind of crazy to me because it's honestly easier to make new yeasts, but we're in an industry where like we're not using hop varieties from 50 years ago. We're
True.
certainly not using barley varieties from 50 years ago. Like the hands of science have really, really changed the other key ingredients in beer, but strangely enough, yeast has kind of been left behind. And now the Renaissance is happening and things are moving, but we've been in this funny spot where it just didn't for a long time.
Yeah. Yeah, that's a good point, right? I mean, everybody knows the Chico yeast strain, and I think that might still be the most brewed, most used strain in North America. I don't know for sure, but that's one of those strains exactly that you're talking about that was used 40 or 50 years ago, and everybody's still using it, and it's kind of like, okay, why? I mean, yeah, we think that we get all these crisp and clean flavors, but now with hops and different hazy IPAs and things, I mean, maybe Chico isn't the best for those.
Exactly. And then we do see the platforms, that I'll call the preferred platforms shift. We have seen Chico give way to the London, I'll call it the London 3 family for, hazy IPAs. And, we are starting to see that give way to lager yeasts, lager yeasts being used in a broader range of applications. So, you do have these undercurrents, but at the end of the day, it's like the last thing I want is for us to all decide that we're going to use a lager yeast from the 50s. Like, we can do better.
Yeah, we can do better. Exactly, we can do better. And so let's talk then about the process. What is traditional yeast breeding, and how does that work? I mean, what are you trying to do in those processes?
Yeah, I mean, it's really just starting with having some kind of idea of what we want to achieve, and then picking parental strains that can hypothetically, meet that requirement. So there is a bit of a mix of science and art at this point, just knowing the strain and understanding it and, what do we like, what do we not like? There's definitely some personal bias in there. I'd love to get to a place where we can just like plug data into an AI and it just says like, hey, take this and this and you've got a new strain. We're not there yet. So what we tend to look for right now is we might look at parental strains in terms of, what can they do? Do they make phenols? What's their attenuation? What kind of aroma compounds are they capable of making? How is the biotransformation, haze, et cetera? And then, if we're looking for a combination of those traits and one parent has the biotransformation, but the other parent has efficient fermentation, okay, let's combine those two. Or maybe we have a strain that's phenolic and a strain that's non-phenolic. Well, we can breed those two and we can actually select for a non-phenolic hybrid, which then can help reveal some of the interesting traits of those phenolic strains. So
Yeah.
we have to know a little bit about the strains going in, but oftentimes it is a little bit arbitrary of like, well, I like this strain, I like this strain. And you do see that in some of the strains we develop. There are some pretty common parental strains that show up just because we like them. We know that they have a lot of genetic potential.
Yeah, it's kind of like it's kind of like they have this they have this ability that you like, or they have this trait that you think might be useful for a hybrid. But it is also, like you said too, you have to understand that yeast, right? You have to understand, is this yeast going to pass on that genetic material, right? And is this, does it have the right genetic material to give to a hybrid? What do the hybrids look like? But okay, so you're kind of, I understand there, you got to sort of start by finding a problem, identifying a gap, like you said, and then look for some candidate parents that might have the genes that you might want to mix together. And one question I wanted to ask real quick is if you have, let's say, a yeast that is a high attenuator and another yeast that is good at biotransformation, why can't you just pitch both of those yeasts in your beer?
You totally can, but the problem with blending yeast is that it's not stable. So, especially these days where cost is a lot more important to brewers. Um, if you're using a blend, typically it's not going to be stable from generation to generation. So then you're kind of stuck paying for that fresh yeast every single time if you want consistent results, which you can do, but I find most brewers are kind of moving away from that one and done approach to yeast management. And so hybrids are a way to accomplish that, without having to be like a one and done yeast blend.
Yeah, and that makes it. And I see what you mean by stabilization is like if you've got a 50-50 blend on your first batch, when you harvest that yeast, it might not be 50-50 anymore. It might be 55-45 or 60-40 or who knows. Yeah.
And we've looked at this with different strains, and it does come down to like if there's a difference in the growth rate or the flocculation, then that population's going to like skew over time.
Yeah, it's going to skew over time. So this is where hybrids come in. Okay, well then, all right, let's say we've got our problem. We've got our two parent strains. How do you make hybrids? How do you breed these yeasts together?
Yeah, so there's two main ways that you can do yeast breeding. There are some more esoteric methods, but we can talk about the two main methods that have been used for most of the yeasts that we make and that I'm aware of in the industry. So there's two different ways that you can do yeast breeding. The first one that I'll talk about is called spore to spore mating. So that's pretty much exactly what it is. I got to explain a little bit of background here. Yeast sporulation. So yeast forms spores, which you can almost think of as like more stress resilient yeast cells that have entered dormancy. Just like a plant makes seeds to survive, for example, over the winter in the soil, yeast will make spores to survive in low nutrient adverse conditions. So it's a survival mechanism. It helps the yeast to ensure long-term survival. What's also really cool is that sporulating and then either breeding or germinating those spores also facilitates genetic diversity because yeast can breed in these conditions. So it's actually a way for in the wild the yeast to create new genetic combinations to ensure its survival because maybe hybridizing is a way for that yeast to survive whatever adverse conditions it's facing, right, in a changing environment. So that's like kind of the natural state. What we can do is we can basically hijack that. So spore to spore mating, you basically starve the yeast to make it sporulate in a culture. So you do the opposite of what you would do if you're making quality yeast. You give it a really, really limited amount of nutrients, and it forms spores, which are these really they look really cool. It's like a little sack of four different tiny cells. And then for this method, you do need specialized equipment. So you use this thing called a spore dissector. It's a fancy microscope that also has a tiny needle on it and a joystick. So if you're like really into gaming, you probably would enjoy this. I personally find it really frustrating. But you take that yeast strain, you dissolve the spore walls with an enzyme. You end up with a puddle of dissolved spores, and you can actually use that needle. It's so small that you can pick up a single cell, a single spore, and basically plunk it onto somewhere on a grid on this agar plate. And then you can go and pick up a spore of a different strain, do the same thing, stick it on the same coordinates, and you've basically you've matchmaked for yeast. You take one spore from one strain and one spore from another, stick them together, and if you're lucky, a new hybrid grows. So that's why we call it spore to spore. We're literally sticking them together and saying, you're a new hybrid yeast now. So it's actually really like if you're good at that technique, it's really simple and fast. It's definitely a skill. There's some people that are extremely good at it and some people that are not. I am not one of them. But
Yeah.
it's you can make hybrids really fast, really efficiently with this method.
So is that essentially, I guess you're just taking like DNA from one of the parent cells and then DNA from the other parent cell and just squishing it together, right?
Exactly.
And then,
The spores germinate. They do this thing called schmooing where they kind of like the buds kind of form and fuse together. And then the actual genetic material also fuses together, recombines, and you end up with a new hybrid.
A new hybrid. That's cool. Yeah. And it's like you said, since there's like some, it's cool that you can like do this with a microscope. So we mentioned this as like a traditional brewing technique, but there's a lot of modern fancy equipment that even makes this, more specific, right? Yeah.
Oh, totally. I mean, this method's been in around for a long time, like 40 plus years. It's easier now. Like back, I know, I mean, my old professor back in the day was like, back in the day, we had to make our own needles, take a little piece of glass and melt it and stretch it just so. Now you can just buy the needles.
Right, walk uphill both ways in the snow to get to the lab, yeah, with no shoes.
So there's better tools out there. The most popular one that I've seen out there, it's they call it the spore play, which is a pretty bad pun for a yeast mating device.
Sure. Yeah. So cool. All right. So that's one of the ways. So you mentioned that there were two main ways. So this spore to spore is like taking those spores and allowing it to sort of recombine into a hybrid. What's the other way?
Yep. The other one is called rare mating. Um, so it's a simpler process. If you want to do it in your lab, it's easier to do because you don't need any really specialized equipment. You know, most brewery labs have a little centrifuge and they can make agar plates, and that's pretty much all you need to be able to do a rare mating experiment. So the way I like to explain it is that you do what they did in Jurassic Park to keep the dinosaurs from surviving if they get out into the wild. So what you do is you expose the yeast to a chemical that makes it unable to produce certain amino acids. So in Jurassic Park, they did this with lysine. They called it the lysine contingency. Obviously, that didn't work out for them. They didn't think
Yeah.
things through. But you can do this with yeast. If you make two strains that are deficient in two different types of amino acids, then you can basically just take that yeast, starve both of them, force them to sporulate, and then just like stick them in a test tube together for a few hours. And you're basically just relying on the rare occasion that two spores of opposite strains mate. But then you have a method to screen for successful hybrids on the other side. So you don't have to like go and look at like thousands and thousands and thousands of cells to see which one's a hybrid. Because you can just plate it on an agar plate that doesn't have the two amino acids that the two parents can't make, because then only a hybrid is going to be able to grow. So you have this nice screening method on the other end, which really helps make it work. So it's a simple method. It's just test tubes and centrifuges. But it is quite labor intensive. It is pretty failure prone. Like just like Jurassic Park, like those mutations go away and the strains just revert, and then you've got, you know, your wild dinosaur again. So it can be a bit of a pain.
I see. And so that's how, like, so if I understand then, so you starve these yeasts of that nutrient, right? And then you cause them, and they sporulate because they're starved of that nutrient. Then you put those in a test tube together and hope that they mate, but for a couple of hours, and then essentially just take the stuff from that test tube, put it into an agar plate that's also starved of that nutrient, right? And then all right, sorry, but they have to be different nutrients. There we go. I missed a key step there. Like one of them's got to be deficient in, I don't know, I'm going to make up something like an amino acid, right? Cysteine versus, let's say, lysine, the two amino acids.
Perfect.
Yeah. And so A is deficient in lysine, B is deficient in cysteine, and then you put it in the tube. If it mates, then they inherit the ability to make those amino acids from the other parent, and then you put them in a plate that's deficient, and if they grow, you've created a hybrid.
Yeah, you missed a step.
Okay.
You have to turn the lights down low and play some slow jams as well, and then you're good to go.
Yeah, right. Exactly. That's the 3 hours there, the waiting window. Got it. Cool. And so that does sound like something that you could do, just on your own at a brewery if you wanted to mess around with this, right? You could just you can make these plates, and it doesn't really take any sophisticated equipment. Right. But that does sound like it's called rare mating. And so I'm guessing this is rare that a hybrid actually forms. I mean, not that the whole experiment's going to fail. Maybe it will, but it's rare that they're actually going to create a hybrid that has the genetic material that needs to survive. And then I guess one step further, that hybrid is going to have the traits that you wanted from the two parent strains.
Yeah, like it's, one thing to get a successful hybrid, but then you don't know if it's going to be good. So you have to screen, you have to test every hybrid, both make sure that it's stable and also make sure that it's able to achieve whatever targets you're trying to achieve.
Yeah. Are there faster ways to do that that rare mating, or I mean, spore to spore sounds faster. Are there other techniques?
There are other techniques, but not as commonly used. Like you can fully just like, there are techniques where you can just like dissolve the cell walls of the cells and like fuse them together. It's called protoplast fusion. In practice, not very practical.
So I've read this term from you, I think it was on your poster at WBC. What is adaptive laboratory evolution?
Yeah, so it's kind of moving on from hybrids, but it's another way to improve yeast strains. So first of all, the thing that I think is fun is that the acronym is ALE.
Yeah, yeah,
So
ale.
since it's beer, you can do it with a lager yeast if you want. But yeah, adaptive lab evolution, ale.
Then we need to change the acronym, right? Like laboratory, adaptive, growth, evolution. I don't know, something
Got
close.
it.
Yeah.
You might hear people call this forced evolution. That's another term for it. You can think of it as strength training for microbes, like expose them to a challenging environment repeatedly, and eventually random mutations will prevail and, basically change that organism. It's the same thing that goes on in a brewery when they repitch, right? If a brewery repitches yeast for, you know, 15 or more generations, you will start to see like real trackable genetic mutation happening. And that's the same thing here. You can just do it on a more accelerated time scale where maybe you're repitching the yeast every 2 days instead of every 2 weeks, for example. So you can force that yeast to evolve faster. The cool thing is for us, Saccharomyces yeasts mutate fast. It's not fun for a brewer because like it will change if you repitch over time. But for us in the lab, that's really cool because we can, select for beneficial mutations on a relatively quick time scale with this kind of method. So it's another really simple method to improve yeasts. All you really need is a shake flask and a stir plate, basically, like an Erlenmeyer flask and a stir plate, and you basically just like repropagate the yeast a bunch of times, and usually check every so often, every 10 generations or so, to see if it's improved in the way that you want it to improve, and then you can go and select like individual yeast colonies or clones to screen further and see, did we hit our target or not. So it's really flexible because it's a way to basically just do what is done in the breweries, but on an accelerated scale and help strength train the yeast in certain environments. So that might be like high alcohol content. It might be like making it more tolerant of hop compounds or biotransformation or certain sugars or even like certain nutrients.
Yeah. Okay. So that makes sense. You're actually, instead of making a hybrid, by mating, you're just forcing that yeast strain to mutate, which is essentially creating a new strain. But it's not two parents or anything. It's more just like forcing that strain to mutate and adapt to its environment.
Correct. And something that I didn't mention when we were talking about breeding is that a lot of yeast strains are that we use in beer are sterile. They're unable to sporulate. They have no sex life.
Yeah. Yeah.
They can't sporulate, and part of that is just because some of the beer strains are just so far along in what I'll call human-directed evolution, repitching, that they don't really need to. They've been in a consistent environment for so long that they just have lost the ability to, sporulate and form hybrids sexually. So they just only reproduce asexually. And that's true of a lot of the more common yeast strains out there. So like Cali Chico, that tends to be sterile or extremely hard to mate. In fact, a lot of the American brewing strains are like that. Lagers, because they're already a hybrid, they're kind of like a mule. It can be done, but it's really, really, really hard. So there's not a lot of examples of that being done. So that's a case where the lab evolution is really useful because if you have a strain that can't be mated to change or improve the traits, this is a tool you can still use to change or improve the yeast.
Yeah, that's a good point. That's something I think I had heard. It like tickled my brain that I had heard that somewhere in the past. But I don't remember where I heard it from. But yeah, that's a really interesting point. Hybrids are a lot like mules. They're sterile, and even brewing yeast, like you said, Cali Chico is a sterile strain. So if you're going to try to combine that with another strain, it's not going to mate and or form spores. Yeah.
That's our experience, at least. Someone out there can correct me if they found one.
Sure. Yeah. We have feedback. If anybody wants to write in with feedback, I'd be happy to take that feedback. But yeah, so okay, I can kind of see now here. Again, we talked about this like as exploratory and discovery, and it sounds like a lot of this is you don't know what you're going to get. right? Like you put these two yeasts together and you hope that whatever comes out is a usable strain.
Yeah, you got it. I mean, even with the forced evolution, you're really just selecting for random mutation, and maybe that mutation that makes the strain more alcohol tolerant also makes it produce a really horrible off flavor, right? Because that yeast is more fit in that environment, but it might not make a better beer.
Yeah. But and so it sounds like that's a challenge then if you've done this and it achieves one goal but it doesn't make the beer better. But it sounds like you guys at Escarpment have had some successes using these techniques. So I want to take a quick break and then we'll come back looking at some of those recent successes. We'll be right back.
Established in 1995, More Beer has been consistently serving the greater brewing community since the times IPA was expected to be bitter and clear, and there are reasons they've stuck around so long. In addition to their massive product selection and excellent customer service, More Beer has locations on both the west and the east coast of the United States, which translates into fast shipping times regardless of where you live. And when you spend more than $59, shipping is free. When you're in need of brewing ingredients and gear, there's no better option than morebeer.com, one of the most trusted shops on the planet.
Hey, I'm Ryan Reynolds. At Mint Mobile, we like to do the opposite of what Big Wireless does. They charge you a lot, we charge you a little. So naturally, when they announced they'd be raising their prices due to inflation, we decided to deflate our prices due to not hating you. That's right. We're cutting the price of Mint Unlimited from $30 a month to just $15 a month. Give it a try at mintmobile.com/switch.
$45 upfront payment equivalent to $15 per month. New customers on first 3-month plan only. Taxes and fees extra. Speed slower above 40 gigabytes. See details.
Save big money on your next project. Now at Menards. Brighten up your garage or workspaces with LED utility and panel light bulbs from GT Light. GT Light light bulbs are energy efficient, and with their easy adjustable light panels, you can direct light where you need it most. Save big on all GT industrial utility and panel light bulbs. And don't forget to check out our weekly flyer for more hot deals happening now.
Save big money at Menards.
One of the easiest experiments to do as a brewer is to change the yeast strain you're using to ferment. You can make split batches and pitch multiple strains of yeast and then assess them for their different brewing characteristics. Now, Richard, this is kind of that step. Once you've made a stable hybrid, you need to test it, right? To make sure that yeast can actually be used in the brewery?
Yeah, and that's honestly one of the big bottlenecks with the whole process is screening, because you got a lot of work to do. You got to make beer, right? And making beer is a lot of work. So we do try to collect basic stats on the yeast. So that's things like attenuation, what aroma compounds can it make, basic sensory profile, haze, flocculation, temperature ranges, and then of course brewing on a homebrew system and basically just making and tasting beers. We tend to do internal testing before we're giving strains to breweries to try out, because last thing I want is for a brewery to throw yeast into 20 barrels of beer and it's garbage. So we try to make sure that it's good. There's all these scientific tests that we do. But I do have to say, if we make a test beer and the keg doesn't kick, then the yeast is probably a dud. If the keg disappears fast, that's usually a sign that the yeast is going to be a success.
Yeah, that's a good one. That's a good metric. I like that. I remember hearing something similar from the Berkeley yeast guys when they were first getting started. They were talking about how some of those yeast would just the keg would kick really fast and they would know, okay, this is going to be a winner. But yeah, that's cool to hear that you guys are doing the same thing. So you're brewing with those yeasts, and then I guess, looking at their different traits. And so when you talk about screening, what specific traits are you looking at? You're looking for okay, maybe flocculation was the goal with this particular blend, or maybe we're trying to do flocculation and biotransformation, or, is that kind of how you're assessing those yeasts? I mean, I'm sure there's also scientific, looking at the, genetic code and making sure that they are actually distinct yeasts. And
yeah, I mean, the stats that are important to brewers, right? The attenuation, flocculation, does depend on what we're looking for and what we've targeted. In some cases, there are some happy accidents where we've just said, okay, let's actually roll with this because this is useful in this other way. Um, or, just comes down to how do we position this and like what styles is it used in? But yeah, I mean, all of those things, and then the next step is always um, getting yeast to breweries. We have this experimental strain program where breweries can sign up, and they can access basically like pre-release yeast strains. It's exactly what hop companies do. Like there's these numbered yeast strains that you can try out, and the deal is you have to give us feedback. You have to give us data, and that helps us to inform our own decision making about whether a strain should be released. It also helps us to like position the marketing based on what those brewers say about the benefits of the yeast. So it's kind of twofold there too, because it's a lot easier for us to just take what the brewers say they liked about the yeast and then, make sure that other brewers are aware of that when we launch it.
Yeah, right. I mean, yeah, it's marketing forward as well, which is helpful. Yeah, all right. So then we have some examples of some yeast strains that were made using traditional brewing practices that are successes. That you guys have actually made it all the way through those processes. So starting with the breeding, hybridization, or the evolution, and then screening it and picking a yeast. This one actually is an entirely new yeast strain that has the characteristics that we really like.
Absolutely.
Yeah, so let's talk about some of those. So, you have a couple that were made using that rare mating technique. So again, I think this was the one that was like sort of simplest. It's the two agar plates, the test tube, and then the deficiencies that you use to make a hybrid. And two of those are Hydra, and I'm going to pronounce this wrong, but Jotun. Is that right? Jotun?
Jotun, I'm pretty sure that's correct.
All right, excellent. All right, so tell us about Hydra and Jotun. So again, these were made using that rare mating technique.
Yeah, so really just started to dip our toes into developing new strains. That was about 5 years ago now, and released a little bit more recently. Hydra, we really really like. What's commonly called Sactois. We call it Cerberus. Um, Super aroma forward Saccharomyces strain. It was super popular when like I was homebrewing and when we were starting Escarpment Labs like 10 years ago. And then it kind of died off because it has some major issues. It's diastatic. Uh, Number one, it's diastatic, and that just limits its ability to be used in a lot of breweries. And the other strain is Vermont, which is also known as the Conan strain. That's a great strain. It's got the nice like peachy ester, but also has some pretty big limitations. Like it's a strain that came into prominence with New England IPAs, but it's actually not haze positive whatsoever. It's kind of challenging to get a stable haze with that strain. So the idea with Hydra was like, can we make a more aroma forward, haze positive strain? The kind of interesting thing about Hydra is that it doesn't have the diastatic trait of the Cerberus, but it also doesn't have the maltotriose like three chain malt sugar consumption of the Vermont. So it's actually a quite low attenuation strain, which in some ways is not a good thing. But what we were actually finding when we were launching it is a lot of brewers were struggling with this new thing that you're very familiar with, Cade, called hop creep. And having a strain with slightly more limited malt sugar consumption can help them to get more like a higher final gravity without as extreme of a hop creep. So that strain became fairly popular, especially for brewers that are, they don't have as much control over the mash parameters to really, really lock in that fermentability. If they're running on more simple brew houses. It's really a cool tool to if you're limited by your brew house, still be able to make those, really, really kind of juicy on the sweet end IPAs and also have a really nice kind of good biotransformation and aroma forward profile otherwise. So really a nice option for brewers still today making hazy IPAs. It's stayed quite popular for us.
Yeah, that's really interesting. And, even as a hazy IPA, I mean, that body and mouthfeel is super important, right? So even like a little bit of low attenuation actually might be really nice and helpful in a juicy IPA, and that makes sense, right? You won't want to use something like Cerberus because of its diastatic nature. And that's really just going to dry out that beer, and you're going to get, lose a lot of that. You're going to have a lot higher alcohol concentration, and you're not going to get that nice aroma and flavor that you're looking for.
Yep, you got it. My pro tip for that one is Hydra and Strata. You just get really, really nice mango. I haven't. I think there might be some engineered strains out there for mango aroma, but I still think that combo is really, really effective.
Nice. Yeah. Hydra and Strata for mango flavors. Strata, of course, being the hop variety. Yeah. Yeah. Cool. All right. And then Jotun, which I'm going to nerd out here a little bit. I'm guessing is like based off of like Jotunheim. Are these two rare mating? Are these related to like Marvel type universe comic books or
yeah, I mean you got to take it right back to the Norse mythology. So yeah,
exactly.
Jotunheim, these like mythological beings in Norse mythology. Say that 5 times fast.
Yeah, the frost giants.
Frost giants, exactly.
Yeah.
So yeah, we named that one because it's kind of a beast of it's a Norwegian beast, basically, is how we describe that. It's actually kind of a scary strain because we hybridized the saison yeast, like a French saison yeast, with a Kveik yeast. It's like a Hornindal Kveik. Because the whole idea there was, and that actually came from a brewer customer of ours who was having issues with, turning around saison. They were comfortable with diastatic, but they were having issues with turning around their saison fast because saison strains are notoriously non-flocculent, right? So it takes a long time to get them to settle out. So we were just like, well, Kvike is really, it's got the aroma production, it's got the temperature range, but it's also flocculent. So let's breed the two together, and that's basically what we did. So Jotun's cool because it's, to my knowledge, one of the only beer strains in existence that has flocculation and that like saison yeast profile and diastatic trait. So, you can make a nice, flocked out saison style beer quite quickly with it, which is pretty cool.
Wow, that's fun. Yeah, because that's right. Because saisons they're so well known for the flavors that the yeast gives, right? And of course, like you said, they're really dry. The exact opposite of what we were of the problem, with Hydra. You want that diastatic, but you also need that flocculation so that you can settle them out and get the beers out of the tanks and into people's hands. Because I see a lot of like saisons, especially being bottle conditioned, and you look at the bottle or breathe on the bottle and everything just goes poof, back up into
great yeast for bottle conditioning. Saisons or sours too. It's like super stress resistant. I mean, I will say this is like a super niche strain, but those
Sure.
that like it really like it.
Yeah, that's a fun one though. That's a fun one. I think homebrewers would especially like that one too, because it's got a lot of characteristics that would be nice for homebrewers as well, in terms of moving saisons through the brew house.
Yeah, just make sure you clean really well after.
Yeah, exactly, because that diastatic's going to stay around. Exactly. Okay, so those were a couple of examples of rare mating. And then you also talked about the spore-to-spore mating, and you've got a couple of examples of yeast that were made with that process too.
Yeah, so the first one we can talk about is Thiol Libre. So, we were a little bit late to the thiol craze. I think Omega beat us to that one, but we did have this in development at around the same time. But it's I think the cool story here is it's a completely different approach. So if you look at strains like Cosmic Punch or Tropics, those are London 3 strains that have that the enzyme that helps the yeast make thiols overexpressed. You've covered this in other podcasts.
Yeah.
So Thiol Libre was kind of a different approach. And so we actually teamed up with Christopher Krogris at VTT, who, to me is one of the, most impressive and prominent beer yeast researchers out there. So that was a really fun project to be able to do. And that was one of the briefs was like, number one, can we actually hack breeding and make it more efficient? And then number two was, can we develop strains to um, improve certain traits like thiol release? So this one's kind of cool, the thiol libre, because we went in and we looked at all of the yeast genome sequences that were available, which was, more than 1,000 at the time, and just looked for that gene, the IRC7, the natural yeast gene that helps it to release thiols. I just looked for like which yeast out there has this gene intact and has the most likely expression or copy number of that gene. And what we found was that there was a wild yeast that was originally isolated from a guava in the Philippines that just so happened to have the most basically natural potential for thiol release. So it's also cool how, we keep running these cases where like science imitates life, where, yeah, of course, a yeast from guava is going to produce the most guava flavor. Guava flavor is thiols. It just makes sense, right? Challenge with that yeast is it doesn't make beer. It's maltose negative. It doesn't do anything in beer. It has no useful traits in beer, right? It just has that high copy number of that thiol release gene. So, we've also done a lot of work with Kvike yeast and, really are big, supporters of what Kvike can do for brewers in terms of efficiency. So, and the other thing with Kvike is that they're easy to breed. So we bred a Kvike strain with this wild guava yeast, and that's essentially how thiol libre got produced. So, leveraging some of the efficient fermentation of the Kvike. The parent strain we picked is also actually decently thiol active. And then we ended up with a yeast with a really high copy number of active IRC7 for thiol release. So it's quite a decent strain for that. It's different from some of the engineered strains, both in terms of background and in terms of how it was accomplished. But it's just a different option that can be used for thiol release. And in particular, the strain's quite good at making the 3-MHA ester. So that's the guava passion fruit ester. Because you have ester production from the Kvike interacting with thiol release from the wild parent.
Yeah. That's amazing too from the wild parent, because I would a lot of people talk about with wild yeast is the phenolic off flavor, right, happening in there too. And that's anytime I hear of like, oh, phenolic, or wild yeast, I'm thinking like, oh no, you're going to get, cloves or, glycols or that kind of stuff. But it sounds like that didn't come through in this, or maybe the wild parent didn't have that. I don't know, but.
Well, I will say the original iteration of thiol libre did have a little bit of POF, phenolic flavor. So we did have to then go do a little bit of further selection to select a non-phenolic clone of it.
I see. Yeah. Oh, that's right. Because again, this is another cool thing about like the exploration is you don't know which genes are going to be passed along, right, to the hybrid. So it might pass along the phenolic off flavor from the wild yeast, but then you can actually find a hybrid out there that didn't have the phenolic off flavor, but still maintains all the other characteristics that you're looking for.
Yeah.
That's cool. That's fun. Okay. And then another one that was also with spore-to-spore mating was Elysium. This one had a little bit of a different goal that you guys started out with, right?
Yep. So Elysium is a hybrid that was released more recently, like last year. Again, you see some of the same characters show up because they're easy to breed and we like what they can do. So Elysium also has a parent of a Kvike strain as well as a really aroma positive Sax cerevisiae strain. So the Kvike strain that we picked has really high production of ethyl hexanoate, which is, we call that just the pineapple ester. And so did the other parent strain. So the idea there is that there's this phenomenon called hybrid vigor, where if you take two, doesn't have to be yeast, it can be corn, take two organisms with a trait and you breed them together, you can get offspring that have that trait amplified rather than just being the same. So that was the outcome with Elysium, where it has what we think is hybrid vigor for the pineapple ester. So really kind of a targeted strain. It does have the Kvike background as well as Sax cerevisiae background. But if you're looking for that pineapple ester, it really is a nice option there. And without it is sometimes when strains have really high production of pineapple ester, it's also part of fatty acid metabolism, so they can be kind of like goaty. Like Brettanomyces is the classic example. Like, yeah, it makes pineapple, but it also makes those like goat cheese flavors, right?
Yeah.
This one's pretty clean with that. So we've had some pretty interesting like hoppy beers or fruit beers with the Elysium, and again, because of the Kvike background, super compatible with home brewing. Really, really wide temperature range, really, really flexible strain.
Yeah. And again, this is an example of you're using a targeted approach, right? You're looking for that ethyl hexanoate, that pineapple ester, and so you're using traditional breeding practices. You could identify the genes that are responsible for ethyl that it doesn't necessarily, there's a lot of steps in producing esters, right? That my main thing here is it'd be very difficult to find a gene that produces ethyl hexanoate because there's a lot of steps that go through that. So you'd have to target a lot of genes to genetically engineer something like that.
Yeah, there's some quite good research on that, and making that happen in like sake yeast. But if you go and do that in beer yeast, sometimes you can get some strange results, like that kind of like goat cheesy flavor. So in our case, hybridization worked pretty well.
Yeah, and that's cool. That again, you've got a success here because you're trying to do that hybrid vigor. I like that. That's something I'll take away from this episode is like two parents that are both strong in a trait, and then you get a hybrid that's stronger, right? It's like athletes having children, right? And then their children
it's
are
unfair,
super athletes,
right?
Yeah, exactly. All right, cool. So again, those two, Thiol Libre and Elysium, using that spore to spore mating technique, and then you also had a couple from that adaptive laboratory evolution. So again, that's where you're taking those yeast and then causing them to mutate. In nutrient deprived conditions or specific, environmental stressors, forced evolution.
You got it. Yeah. So the first example of that is our crispy. And yeah, I do want to give full credit for basically all the lab evolution ideas to former colleague Eugene Fletcher. He's a great yeast scientist. He was with our team for a couple of years, and now he's a professor in Ottawa. So he graduated to the big leagues, which is awesome. But yeah, he came in and basically was like, "Why aren't you guys doing lab evolution? It seems like it would work to improve your strains." And so he just started doing it, and that's where a lot of these came from. So crispy started as a blend of strains, which, as we've talked about, tend to not be very stable. So when brewers were repitching it, they would get unexpected results. It would be like hazy, or the attenuation would change. A lot of issues. So, and the problem there was that like the two strains, we kind of needed both in the mix. One was hitting the right flavor notes, but wasn't very attenuative, and the other one was correctly attenuative. So what we ended up doing was we took the less attenuative strain and we evolved it over time in high gravity wort to basically make it better at attenuation of the wort, better at consuming some of those more challenging malt sugars, and ended up with an evolved strain over about 48, 47 of those basically fermentations or propagations. So it kind of solved that problem because we ended up with a single strain that is stable when it's repitched. And, at the time, this was 2021, at the time, we were at the peak of Kvike pseudo lagers. I think it has kind of died down now in favor of just lagers, normal lagers, but still has its place. I mean, it can be quite useful, especially if you're under production time pressures. And certainly as a clean strain to get beer fermented fast, it works really, really well.
Yeah, I mean, a fast fermenter, a high attenuation, and flocculent, right? I mean, that's as long as there's no crazy esper production or something, that'd be a big one for brewers to be able to use, is make a clean beer that ferments quickly and clears up, that you can get it in and out of the tank quick.
Yeah, and you can make it not taste like Kvike if you start it cold and let it ramp up after a day or two. So that's also my pro tip. If you've used Kvike and you got that like acidic twang, try starting it like 5 degrees below your target temp for a couple days and then ramp it, and it'll be way more neutral, way less acidic.
Nice, nice. That's a nice pro tip to remember. You're dropping bombs here. You're dropping tips all throughout, which is great. And then the other one that you guys that we were going to talk about for this episode is terps. That one's cool too. Tell us about terps.
Terps, yeah, man. I want more people to use this one because it's honestly, I'm super excited about this strain. It's an American ale strain, so like a California style ale, that was lab evolved in the presence of toxic amounts of terpenes. And the idea there was like, well, if we make it deal with all these terpenes, we're basically going to have to, select for improving its terpene or altering its terpene metabolism. And maybe that will lead to improved terpene biotransformation. That was a total shot in the dark because, it could have gone down the other path and just consumed them in a different way. But in our case, it did work. So, the main terpene biotransformation is that typically we see in from yeast is geraniol, which is that kind of floral aroma in hops. Yeast will convert to citronellol, which is a citrusy lemongrass aroma. It's, to my knowledge, not naturally found in much abundance in hops. So it's a really good signature of, what can yeast actually do to hop compounds, seeing that biotransformation. So basically expose the yeast to high levels of terpenes and, hoping and praying that it would improve its terpene biotransformation. Lucky for us, it did. This is one of those cases where it's pretty hard to engineer. You can get yeast to, for example, make linalool or geraniol. There's products out there for that, but actually getting that citronellol transformation, we don't really understand exactly what genes are responsible there. So it's a great target for the lab evolution, right? And so the end result there is that it's still an American ale strain, with all those benefits of like high attenuation, efficient, all that good stuff, but with more terpene biotransformations. So usually what I recommend to people with that one is like if you have a hop that you're stuck with, or that you just think is a little bit too dank, try it because that's always, to me, the classic benefit of terpene biotransformation is you can take those like floral or dank notes and turn it into something that's more citrusy. And, in general, that's what most consumers are looking for. So, if you're stuck with a lot of Columbus or Mosaic that's just a little too dank, then it's a really good option to use a yeast like this to correct that.
How fun. Yeah, that's interesting because I don't normally think about like using yeast to solve issues with hops, right? Or to do that. But that's a really good application there for terps. Very cool. Yeah. And then, so we started at the top of the show talking about, how like these techniques, we were talking about genetic modification, genetic engineering, and traditional breeding. And we talked about like having different tools in your toolkit, right? And how they can all be used to create, kind of in parallel paths to create some cool, yeast strains or to create some cool outcomes that brewers can use. Well, you can also use combinations of traditional breeding practices to make a strain, and that's where this new strain Pomona comes in.
Yeah, you got it. So that's the newest one, at least in terms of its current format. So we've released that in partnership with Lallemand Brewing. So the really cool thing there is that it's a dry yeast. You can get it all around the world. So if you've been excited by some of the strains that we're developing, but you can't get it because we're in Canada and you're in New Zealand, now there's a solution for you. So that's been really, really exciting to partner with them to make this strain available. And I also think it's this strain is a good culmination of all the things we've been talking about. Really, we wanted to create a new yeast for IPAs, a new platform strain, just because of all the things I've talked about, how we don't really have anything new as a platform strain. And, I really love for brewers to have another option beyond, your Cali and your London 3 and your now 3470. So that was the goal. And I think we've achieved it. It has some pretty unique and distinctive characteristics. So, we ended up with nice kind of peachy, stone fruity aromatics, just, a different profile from what's conventionally out there, which I hope is useful to brewers. And that is made through both breeding and lab evolution. So making a combination of strains, and then putting it through the lab evolution to basically make it better at the flavor development in IPAs and better at the haze production in hazy IPAs. So, when I talked about like stacking methods, this is a good example of it.
Yeah, and it's cool because it blends all of these characteristics, right? I think one thing too that you mentioned to me while we were talking about it is this is a strain that can be used from session pale ales to triple IPAs. So the alcohol tolerance on it is huge, and so this is from a blog post that you recently wrote about it, but I think it summarizes what we've been talking about on today's episode. So it says we aimed to create a strain with the right ester and terpene transformation profile. We got the esters from our mating of two strains, one aromatic and one efficient, but found the hop transformation of the hybrids to be lacking. So we devised an adaptive laboratory evolution strategy to improve the candidates. We adapted the hybrids in New England IPA wort full of hop compounds to adapt to. The resulting adapted hybrid we chose is Pomona, which shows a lively balance of esters and terpene biotransformation that yields a delightful peach aroma in IPA style beers. And so, of course, you took an aromatic strain that had the esters that you wanted, an efficient strain that was able to stand up and into these like triple IPAs, for example, with high alcohol, and you bred those together, didn't like the candidates, devised an adaptive laboratory evolution to say, okay, let's put them and make them interact with these hop compounds, and the result is this great yeast that can be used from a session pale ale all the way up to a triple IPA, and as you said, potentially a new platform style yeast, one that you can use this in any IPA as your platform to build on.
Yeah, that's what I hope. I mean, that's really the goal there, is not just one specific trait, which is that's awesome too. I actually, I love some of the strains that do one thing really, really well, but it's nice to have a strain that can do a bunch of different things and really be this platform to layer different flavors in. And I honestly do think that it pairs really well with some of the hops that are in development and some of the styles that are in development. Like as the hazy IPA starts to blur with the West Coast and become a little bit drier and not necessarily as heavy. It's a really complementary strain to that direction, and to some of the new hops out there. So I will plug HBC 1019. That it goes really, really well with Pomona. It's got its own sort of peachy thing. Nectaron, as I said, and then for a public hop, Vista works great too. So gotta plug the public hops too. There's like a lot of really cool new options that brewers can combine and produce new profiles in the beer, because at a certain point, I do think people are going to get sick of Citra Mosaic and London 3.
Right? Yeah, exactly. It's so cool to see like a yeast that's designed for hops, for the new age hops that we're exploring, right? And again, there's thiols and thiol release, and you guys have got a strain that focuses on that. Of course, Omega Yeast has been doing their thiolized yeast strains. Other yeast labs have got that, but this one also is terpene biotransformation. So this is also, that component that we were talking about, looking at, other ways of getting aroma. That peach aroma is an ester, right? Or a terpene that's biotransformed, right? That is a unique flavor that people really like in some of these new age hops, and that's kind of where we're going. That's cool.
Yeah, we actually had to like dig into that a bit with the Lallemand team, because I mean, it's been really cool collaborating with them, because they were a little skeptical. They're like, what do you mean peach? Like, what is this actually? So they actually went and did some additional work and started to look into the lactones, which we weren't able to measure before. So, it is known that peach aroma in wine is a combination of esters, terpenes, and lactones. And so what we were able to see is that there actually is, differences in the lactone profile with Pomona versus other strains. Same thing with the terpenes. Some strains are also good at those aroma productions, but it could be that the specific combination of terpenes, lactones, and esters with the Pomona is helping to give that peach-like impression.
Yeah, right. It may not be just one thing. Again, this is an example of where, genetic editing or gene editing targeting one component wouldn't work. It's where yeast breeding, again, shines in this way, at least, again, on this path, right? And so we've just looked at all of these successes, and we talked about Galaxy Brain. And of course, whenever I think about Galaxy, I think of Star Trek, which is one of my favorite shows. So to make a Star Trek reference, it sounds like you guys kind of are on this like 5-year, maybe longer, but a deep space mission, boldly going where no one has gone before and doing all these things to come up with creative new yeast. So do you have, what's next on the horizon? Just continuing to explore?
I think we need to make a tricorder for yeast. I'm being honest.
There we go.
I'm not kidding. That's pretty much the goal. Like if we can have a simple and fast method to learn everything we possibly can about a yeast strain, so that then we can first of all empathize with it and understand it and then improve it, breed it, adapt it with less guesswork. That really opens up a lot of doors.
That'd be cool. Cool. Yeah, a little scanner, just like scan it and then say, okay, you scan this yeast cell, and then here you go.
Yeah, sequence the genome, get some aroma data, get some, phenotypic data, and use some fancy math to tell us what we can do with it.
Exactly. Well, the AI is probably going to do that for us here in the future, but that's cool. That would be fun. Well, all right. So we talked. This was a great episode on traditional breeding. We talked about a lot of the different practices that are used, some successes that you guys have had in this. If you want brewers to take away just one thing from today's episode, what would it be?
Yeah, if you thought the last 20 years of hops was fast and furious, yeast is going to quickly eclipse it. It can develop so fast. There's a bunch of different companies competing and working on this, so it's going to happen really fast. So, my take home is keep up, because new yeasts are going to solve long-standing industry problems. They're going to open up new ways to make beer, and I think a lot of people are kind of burying their heads in the sand right now and not trying new things. I encourage you to do so. Keep experimenting, because we need to experiment to find the next big thing and thrive as an industry.
I love it. Keep up. Keep brewing with the new yeast strains. Keep checking them out and trying them and see what you can do. And yeah, it's interesting. I wonder if we'll start to move away from, Chico and London 3 as we start to get new yeast strains out there that can solve other issues. Yeah.
I mean, people are making a lot of lagers, and I'm here for that. I love lagers, but it's all about diversity and having lots of options.
Totally. And I mean, we didn't talk about necessarily lager strains today, at least as far as I know, but there are options, I'm sure, for hybridizing cold tolerant strains in the future, which would be cool to look at. Well, Richard, this was fun. Thank you so much again for joining me back in the Brew Lab, and we've got to get you back before another 130 episodes go by.
Let's do it.
All right. Well, listeners, in the show notes, you're going to find links to a blog post and a YouTube video about yeast development, including a lot of the stuff that we talked about on today's episode, and also a link to a WBC poster, a World Brewing Congress poster that Richard shared just recently up in Minneapolis. And of course, next week, Jordan and I will be back applying the science of this week's episode. See y'all then. The Brew Lab is a production of Brewlosophy, where they who drink beer think beer. Don't forget to visit brewlosophy.com to read about our weekly experiments and other brewing adventures, and listen to us talk about it on our other show, the Brewlosophy Podcast. Thanks to all of our sponsors and patrons that help make this show possible. If you'd like to receive a reward for helping us do what we do, visit patreon.com/brewlosophy to see how you can do just that. Thanks so much for listening. We'll be back in the Brew Lab with another guest next week. Until then, think beer.
Hey, I'm Ryan Reynolds. Recently, I asked Mint Mobile's legal team if big wireless companies are allowed to raise prices due to inflation. They said yes. And then when I asked if raising prices technically violates those onerous 2-year contracts, they said, "What the f*** are you talking about, you insane Hollywood ass?" So to recap, we're cutting the price of Mint Unlimited from $30 a month to just $15 a month. Give it a try at mintmobile.com/switch.
$45 upfront payment equivalent to $15 per month. New customers on first 3-month plan only. Taxes and fees extra. Speed slower above 40 gigabytes e details.